Morning, everyone.
Good morning.
Good morning.
Welcome back. Good morning.
Good morning.
Hi, Rachel.
Good morning.
Good morning. Welcome back, everyone. Hope you're doing well. It's nice to see you, quote unquote, in the virtual world, in the cyber world. So welcome back as we sip on a cup of coffee and rub the sleep out of our eyes, right? 8 o'clocks are difficult. Okay. Well, folks, today we are going to finish our coverage of chapter 2, which is the chemistry chapter, as you know. So that's what we're going to be focusing on this morning. Before I jump back into chapter 2, do you have any questions you'd like to ask about anything you've been reading or studying or thinking about? Okay, well, please let me know if something comes up, right? That's my job. I'm here to try to help you. Okay. So Kelly, if you have a question, go ahead and ask it. I'm just looking at the chat window at this time. Yes, Kelly, the homework was on the skeletal system. That's correct. Okay. All right. I'm going to get started. Kelly, there'll be many times where I ask you to work ahead of me. Okay. So yes, those things are due today. Okay. Kelly, I'd encourage you to come chat with me during office hours if you're confused about things. Okay. So I'm going to share my screen with you. Okay. And here's about where we were last time, where we were talking about the important biological properties of water. And the third statement here is that water is a fantastic temperature buffer. Water is really good at resisting changes in temperature. And this is important because it ensures that our body, which is predominantly made of water, gets to stay a relatively constant shape, okay? And this statement here, changes in temperature, change the shape of molecules, which changes how the molecule function. It's such a very, very important statement that, you really should memorize this and keep this in your memory banks, okay? Any environmental condition that can change the shape of a molecule, whatever that condition is, in this case, temperature, any environmental condition that will change the shape of a molecule will change the function and behavior of that molecule, okay? That is the reason that you feel so poorly when you get a fever. Think about it. If you have a fever, if your fever gets up to 103, you're not feeling too good, right? And it's largely in part because the molecules of your cells, of your tissues and organ systems are struggling to maintain a correct shape to function properly, which is why you feel so poorly. Okay, we're going to continue our conversation with water. Do you have any questions so far about the statements that have been made? Okay, all right, gang, so here we go. Number 4, continuing to talk about the important properties of water. If you want a page number in your textbook, the pages that talk about water are somewhere around pages 40 and 41. Okay. And so bullet point number 4, water is a wonderful lubricant for the body. By being a lubricant, what we're saying is that water has the ability to reduce the friction between structures that are rubbing up against each other. Okay, think about even in your study of the skeleton in lab, right? You're thinking about the various joints like the shoulder joint, the hip joint. Think about how the bones that represent the interface of these joints are rubbing against each other. Think about how the head of the femur locks into the acetabulum of the pelvic girdle, okay, and they're going to rub against each other as you rotate your leg, as even you move your leg to walk across the floor. It's good that in that space there's a special substance called synovial fluid, which is made out of water, which reduces friction, so we reduce inflammation of the joint. Okay, so hopefully that is a reasonable statement to you. Water is able to reduce friction and minimize inflammation that occurs when structures rub against each other, when structures inside the body rub against each other. Okay, so water is very good at reducing friction. Water is a wonderful lubricant. All right. Next talking point. Now, this is basically the four points I put into your notes here are basically things that your textbook gives you. I'm going to mention another one in this context, and that is number 5. Water is able to influence the pH of a cell. Okay, water is able to influence the pH of a cell and or body fluid. And this one may not immediately be as obvious to you. So the first thing I want to say is how? How can water influence the pH of a cell or body fluid? Here's how. Water influences the pH of a cell or body fluid by acting as the solvent that dissolves in acid or base. Okay, by acting as a solvent that dissolves in acid or base. Okay. All right. So I'm going to put number 5 there as an important biological property of water, that it is able to influence the pH of the internal environment of your body. Again, it does so by being the solvent, the liquid that dissolves the acids and bases. And this is going to open up a brand new talking point, a major talking point for us here today, where we need to talk about pH and what it is, how it's measured, and why it is important. Okay. So this is our next talking point, is the topic of pH. All right. So I'm going to make this a new heading here. pH, acids, bases. Okay, that will be the heading of our next conversation here. Okay, so we're going to have to start our conversation by defining some terms, defining some words for you so these things make sense. Okay, so first off, let's define pH. pH is a scale. Okay, I'll say it this way, it's a scale, then I'll come back and talk about it. A scale used to measure the hydrogen ion concentration of a liquid. pH is a scale used to measure or represent, if you will, the hydrogen ion concentration of a liquid. Okay, now don't let the word scale throw you off here. Okay, think about it. I mean, we can think in practical terms. In your bathroom, you might have a scale. You might have a bathroom scale that you put on the floor, you step on it, right? And it tells you how heavy you are, what your body weight is. You might have a scale in your kitchen that you use to measure out portions of food for a recipe. So that's a scale. If you think about a yardstick, you might have a yardstick in your closet at your house. You can get that yardstick out and measure how tall your children are, or how long your furniture is, or the length of anything you need to know that information about. So the yardstick is a type of scale. It's an object that allows you to measure things, to represent distance. pH is a mathematical and chemical scale, a logical scale used to represent the hydrogen ion concentration of a liquid. Okay. So don't be worried by that word scale. It's simply a way to measure something. The other word in this definition that I find students sometimes have trouble with is the word concentration. So we better talk about concentration as well. I'm going to ask you to contribute in the chat window. Okay. All right, here we go. What is the definition of concentration? Folks, what is concentration? How should we interpret this word so we can interpret the definition of pH? What is concentration? What does it refer to? Does anybody have any experience with this word that they can contribute to the conversation here? Okay, thank you, Brittany, for your definition. And you know what, Brittany, that is a definition of concentration. Brittany says the power of focusing one's attention on something. That is a correct definition of concentration in a different context of using the word. You're right, Brittany. It's not how we're going to use it in our course, although I would argue as your teacher, I appreciate your concentration when you're studying. I appreciate your concentration when you're taking an exam. Okay. So just take a look. Please repeat.
Is it adding water?
I'm sorry, I'm not quite hearing the entire statement you made there. Please type into the chat window there and I can verify what you're saying. Okay, so as everybody looks at the chat window, you start to see some of the relevant answers for sort of the sciences, the biology, right? The amount of one molecule in a substance, okay? The amount of substance per unit measurement. These are the things that I would focus on here. And so Kathy, you mentioned about adding water. Certainly water is part of the definition, okay? So let's define concentration this way. Concentration is the amount of a substance. And when we talk about an amount of a substance, that's typically best measured by a mass, okay? A mass, in other words, 5 grams, 5,000 grams, 12 pounds, 4 tons, whatever it might be, an amount of a substance. That substance is measured in a unit of mass, like grams, dissolved in a volume of liquid. That volume could be any unit of measurement. It could be milliliters. So as an example here, okay, the average blood volume of an adult male is 5 to 6 liters. The average blood volume for an adult female is 4 to 5 liters. So for our conversation right now, let's just say that the typical blood volume of both men and women is somewhere around 5 liters, okay? Is approximately 5 liters. Okay, so now, if we had an adult male or female with a blood volume of 5 liters, and that adult swallowed 1 tablet of baby aspirin, 1 tablet of baby aspirin, which maybe you have some at your house. Typically, baby aspirin comes in tablets that have about 81 milligrams of aspirin in each tablet. So my point here in the context of this conversation about concentration is that if that person swallowed a tablet of baby aspirin, the concentration of baby aspirin in that person's blood volume would be 81 milligrams of aspirin dissolved in 5 liters of blood. Okay. Okay, if a person swallows 1 tablet of baby aspirin, then the concentration of that aspirin in the person's blood would be 81 milligrams of aspirin dissolved in 5 liters of blood plasma. Okay, this is a statement right here. 81 milligrams of aspirin dissolved in 5 liters of blood plasma is a statement about concentration. This is a concentration statement. Okay, folks, let me ask you, is concentration making more sense now that I've given you this example? Is there anything you'd like to ask about concentration? Okay, well, let's come back here to the topic of pH, right? How does, when we talk about pH being a scale used to measure the hydrogen ion concentration, how do we apply this example for pH? So the example for pH here is that when we're talking about the hydrogen ion concentration, we're saying that there's a certain number of hydrogen ions dissolved in a certain volume of water. Okay, a certain number of hydrogen ions dissolved in a certain volume of water. Now, of course, hydrogen ions do have a mass, but we're not going to get into that right now because that's really intimate chemistry, talking about Daltons and things. But the point is, if we have a certain number, arguably of a certain mass of hydrogen ions dissolved in a certain volume of water, that would be what pH is. pH is the hydrogen ion concentration. Okay. All right, so let's continue here. Some other terms that we need to talk about: acids and bases. An acid, what is an acid? An acid is any molecule that, when placed in water, releases hydrogen ions. Okay, any molecule that, when placed in water, releases hydrogen ions. An alternative and equivalent definition would be this: any molecule that, when placed in water, increases the hydrogen ion concentration of the water. I'm going to give you an example in a moment. Kathy, I see your comment. I understand. It's okay. So an acid, any molecule that, when placed in water, increases the hydrogen ion concentration of the water. In other words, the number of hydrogen ions in that water goes up. Okay, so let me give you an example here that maybe will make more sense of this. And it's just easier for me to draw sometimes, so I'm going to draw this a little bit. Okay. And so here we go on this. Okay, so let's talk about hydrochloric acid. HCl is short for hydrochloric acid. And you say, well, that sounds like powerful stuff. It is powerful stuff, but you know what? You actually have hydrochloric acid inside your body, inside your stomach, inside your belly, right? Hydrochloric acid is one of the primary ingredients of your stomach juices. If you have your textbook open, I hope you do, take a look at table 2.4. Table 2.4 on my physical textbook, it's page 44. It may not be page 44 in your digital book. It might be in that vicinity, but table 2.4. So let's see if you folks are able to find this from your textbook, table 2.4, in the chat window. What is the pH of gastric juice? What is the pH of gastric juice? Okay. Hey, thanks everybody for contributing. I'm starting to see it there, right? So everybody take a look. If you can't find it, notice that it's 1.2 to 3. In other words, it's a range of values. Okay, so it doesn't have one constant pH, but it's a very low range of values. 1.2 to 3 is the pH. Folks, gastric juice is primarily hydrochloric acid. Okay, that's what it basically is. So you have this powerful acid in your stomach, which aids in the breakdown of materials that you eat, okay, to help with digestion. You're going to get digestion in AMP2. You'll learn all about it then, okay? But right now, what I want to mention to you is an example of how hydrochloric acid works like an acid. So here's the deal. If you have HCl plus water, what will happen is the HCl, the hydrochloric acid, will break down into hydrogen ions, chloride ions, and water. Okay, so now I'm going to ask you folks a couple questions, but first I need to color code some things so you can see what I'm talking about. So I'm going to ask you some questions. First, I'm going to ask a question about the left side of the chemical equation, which I'm drawing a red bracket on the bottom of it. And then I'm going to ask you a question about the right side of the equation. Okay, so now with this color coding, let me ask you a question in the chat window. Okay. So on the left side of the chemical equation, the side where you see red, how many hydrogen ions do you see? Okay, there's the question, folks. On the left side of the chemical equation, how many hydrogen ions do you see? How many hydrogen ions are on the left side of the chemical equation? Okay. Okay. So, all right. Okay, so I'm looking at the answers here. Go ahead, everybody, take a look at the answers being presented forward there. Okay, and just take a look at what people are saying. All right, and I will say that, you know, I'm looking at the responses as well, and there's only one response that I can see that was actually correct. Okay, so maybe this will be a shock to you, but let me say, Raquel, good job, because your answer is correct. There are 0 hydrogen ions on the left side of the equation. There are 0. In other words, a hydrogen ion is an H plus, is it not? It is an H plus. And so what I'm saying is, on the left side of the chemical equation, if I can get my technology to work, there are in fact 0 H pluses, right? You don't see H plus anywhere on the left side of the chemical equation. So there are 0 hydrogen ions. Now with that interpretation, and you know, hopefully as you process what I'm saying there, with that interpretation, how many hydrogen ions would there be on the right side of the equation? How many hydrogen ions are there on the right side of the equation? Okay. Now it's making sense, right? Because you know how to properly interpret the information. You're absolutely right. Good job, everybody. So the answer is now 1. There is 1 hydrogen ion on the right side of the chemical equation. So let's think about this. As we go from the left side of the chemical equation, we put an acid in the presence of water. The acid breaks down and releases hydrogen ions. We went from 0 hydrogen ions to 1 hydrogen ion. Is that consistent with our English definition? Any molecule that, when placed in water, increases the hydrogen ion concentration of the water. We went from 0 to 1. That is an increase. So that is consistent with the definition and behavior of an acid. Okay. All right. Other questions? All right, let's define what a base is then. Let's see if I can do that. If I can get my technology to work here. All right, number 4. Let's see, I think I'm going to bring it down here. Okay, number 4 is the definition of a base. A base is any molecule that, when placed in water, binds to free hydrogen ions floating in the water. Any molecule that, when placed in water, binds to free hydrogen ions floating in the water. An alternative way of saying this is any molecule that, when placed in water, decreases the hydrogen ion concentration of the water. Okay, so it's important that you see that an acid will increase the hydrogen ion concentration and a base will, in fact, decrease the hydrogen ion concentration. Well, let me give you an example of this, again, to provide more context to you. Okay, so a good example of a base, in fact, a very strong base, is a substance which is NaOH. This is the chemical, this represents a substance called sodium hydroxide, okay? Sodium hydroxide. All right. On table 2.4 of your textbook, at the bottom of table 2.4, you can see the last item there is called Li, L-Y-E. Okay, Li is basically pure sodium hydroxide, not quite pure, but it has a pH of 14. Okay, so it's on the other end of the pH scale. Sodium hydroxide is commonly found in substances like oven cleaners. Now, I know today it's probably not very fashionable. When's the last time you spent any time actually cleaning your oven? But if you were so motivated and you wanted to clean your oven, most of the cleaners have sodium hydroxide in. At any rate, here's an example of how sodium hydroxide will behave as a base. Okay, so sodium hydroxide, forgive me, sometimes my tech gets the better of me here. All right, so sodium hydroxide, if it's placed in water where there are some free hydrogen ions, here's what's going to happen. And I'm going to show this in multiple steps. Okay, the first thing that would happen is sodium hydroxide will separate into its individual ions. So sodium ion with the positive charge and the OH ion called a hydroxide ion with a negative charge. Okay, they're going to separate like this. And then what's going to happen, and I have to go below what's going to happen is the OH, let me highlight this, the OH and H here are going to attach to each other because they're attracted to each other because of the opposite charges. So they're going to come together. Okay. And so what that's going to do is leave you with this. It's going to leave you with sodium with a positive charge. You're going to form a water molecule, right? Because OH and H is two H's and an O, H2O. So that's a water molecule plus another water molecule. And if we sum it up, you're going to be left with this, 2H2O. Okay. So let's do the same thing we did here. Okay. I'm going to highlight this area with a red bracket. And then I'm going to highlight a different area here. I'm going to highlight this one right here with a blue-black bracket. Okay. So do you notice that on the left side of the chemical equation with the red bracket, there is in fact one hydrogen ion, right? It's right here. There's one hydrogen ion right here. Okay. Then it goes through some chemistry, and ultimately we end up with the blue bracket, where at this point you can see there are no hydrogen ions. So there are 0 hydrogen ions on the right side of the chemical equation if we follow the process far enough along. So what have we done? We've gone from 1 hydrogen ion on the left to ultimately 0 hydrogen ions on the right, which is a decrease in the hydrogen ion concentration of the water. Okay, so that's how a base will behave. Okay, folks, do you have any questions right now? Okay, sometimes you don't know what to even ask, right? I mean, sometimes you just don't. Sometimes it's not always obvious what you should be asking. So as you study, be sure that you're writing down questions that you can bring into class. You can come see me during office hours. You can go to a tutor with. It's important that you write your questions down and you make an effort to answer them. That's significant. Okay, so next statement. Okay, why does this all matter? Why does this all matter? Okay, why am I worried about pH in the body? Why am I worried about acids and bases? Who really cares? Here's the deal. Okay, just like we talked, I don't know, 10 minutes ago about how changes in temperature can change the shape of molecules. Here's the big punchline. Changes in pH also will change the shape of molecules. Okay. If the environment that a molecule is in has its pH changed, then the shape of that molecule will change and its function will change. Okay, so both changes in temperature and changes in pH lead to changes in the shape of a molecule, which changes the behavior and function of the molecule. Okay, just like temperature changes, pH changes will change the shape and function, meaning activity, meaning behavior of a molecule. Okay. All right. So let me mention a clinical connection here. Let me mention a clinical connection. Okay. Let me ask you a question in the chat window here. Do you know someone that is diabetic? Please give me a yes or no if you know anybody that's diabetic. Okay, As you look across the responses in the chat window, you can see that just about everybody knows somebody that's diabetic. Okay, diabetes is a very common condition, unfortunately, in our society for a variety of reasons. We do have an unhealthy relationship with food in the United States of America. But at any rate, here's the clinical connection to pH. Okay, and I won't get into a whole long-winded conversation here. That would be for another time. You'll talk more about diseases of the endocrine system in AMP2. But here's what I'm trying to bring out here, okay? Often, diabetic patients, particularly when they go off their medication, have altered blood and body fluid pH. Often diabetic patients, particularly when they go off their medication or they're not being treated at all, have altered blood and body fluid pH. So the question becomes why? Here's a quick answer that you may not immediately understand everything I'm saying yet, because you just need more background to understand. When a person is diabetic, their body is confused, if for lack of more precise language right now, their body is confused about how to properly metabolize sugar. And when that happens, typically what goes on is that patient's body begins to abnormally metabolize lipid instead of sugar. Long story short, again, with some things that you don't necessarily know yet, when a diabetic patient starts inappropriately burning a type of lipid inside their cells called triglyceride, the cells break down the triglyceride. The triglyceride, when it gets broken down, is broken down into fatty acids. Those fatty acids are ejected from the patient's cells and put into the patient's bloodstream. So you're taking a fatty acid, putting it into the bloodstream, which lowers the pH of the bloodstream and acidifies the bloodstream. That has long-term negative consequences for the overall health of a person, because you are changing the shape of molecules that the body has to function correctly. Okay, so that's sort of the 50-cent quick explanation of why there is a clinical connection between health and body pH. Okay, so let me just answer the why here in your notes. So fat cells, which are more properly known as adipocytes, will incorrectly break down triglyceride molecules and eject fatty acids into the bloodstream. This acidifies the bloodstream. Okay, so, you may not be particularly interested in learning about the technical details of pH. It may not be something that's interesting to you, but there is reason that it has to be known and understood to treat people successfully for diabetes, because pH will change in diabetic patients who are experiencing the disease and are not being treated successfully for the disease. Okay, all right. Thoughts and questions, gang? Okay, so there's another statement that I'm going to expect you to know for an exam that's important for you, and here's what that statement is. Okay, so first off, let's define the pH scale. Okay, the pH scale is actually graphically kind of shown to you on figure 2.12 in your book. And I won't redraw it, but I'll just make a little statement here. It starts with 0, right? And it goes out to about 7 for neutral. And then it finishes out at about 14, right? You see that on figure 2.12. At the bottom, you see pH values of 0 to 14. Okay, everything between 0 and 7 is basically acidic. Everything that's right around the number 7 would be neutral. And everything basically between 7 and 14 is in fact basic. Okay, so that's how you would apply the languages. So if it's less than 7, it's acidic. If it's greater than 7, it's basic. If it is 7, then it's a pH which is neutral. Okay, so that's what the scale is. And so I want you to appreciate the following statement about the scale. For every one integer change on the pH scale, there is a 10-fold change in the hydrogen ion concentration. Okay, one more time. For every one integer change on the pH scale, there is a 10-fold change in the hydrogen ion concentration. Okay, so what do we mean by this? Here's the deal. Okay, if we compare two solutions, solution A with a pH equal to 5, solution B with a pH equal to 6, this means two things. You can interpret it two ways. This means that solution A has 10 times more hydrogen ions than solution B. Alternatively, you could also say the following. Solution B has 10 times fewer hydrogen ions than solution A. Okay, let's take a look at this again. If you had two solutions, solution A in your left hand with a pH of 5, solution B in your right hand with a solution pH of 6. Okay, so some liquids in a cup in your left hand, a liquid in a cup in your right hand. Solution A in the left hand would have 10 times more hydrogen ions than solution B based on its pH value. Solution B would have 10 times fewer hydrogen ions than solution A. Remember, the lower the pH value, the more acidic. The higher the pH value, the more basic. Okay, so why is it that there's a 10-fold change in hydrogen ion concentration for every one integer change on the pH scale? Why is that? Okay, why is this true? Okay, so now I'm going to get just a little bit mathematical with you. Don't be frightened. I'm not really going to beat you up with heavy math and chemistry on the exam, but I won't say that I won't ask you anything about it either, right? You do have to know something. You need to be thinking about this. So pH is based, pH is, how should I describe it? pH is measured as a base 10 logarithmic scale. pH is measured as a base 10 logarithmic scale. Now, this phrase right here is scaring everybody right now, right? I mean, that's what's happening right now. This is scaring you, particularly if I said I need you to do calculations with it. Don't worry, this is not going to get intense, but at some point in science and in your education, you need to think about technical details that sometimes intersect with math. So let me give you an example that I hope will take a little bit of the scariness out of this. Okay. Let's talk about acid rain. Let's talk about acid rain. Okay, so obviously acid rain is not something relevant necessarily for the inside of your body, but the principle of using pH is relevant here, whether I'm talking about acid rain or something about your blood pH. It's the same principle. So I don't know if it's still considered to be the case, but I know when I was growing up that Los Angeles in California was considered the smog capital of the United States because there was just a lot of pollution in the air. Los Angeles has a reputation of having pretty poor air quality, right? So if we say this, just as an example, Los Angeles, okay, Los Angeles rain has been measured at a pH of about 2.3. That's pretty darn acidic. It's pretty low on the pH scale. Normal meaning average rain across the rest of the continental United States has a pH of about 5.3. Okay, so normal rain is about pH equal to 5.3. So what I'm going to do is subtract the two, and I'm going to take 5.3 minus 2.3, and obviously the answer to that is 3. There are 3 pH values, 3 integer values difference between Los Angeles rain and normal rain across the continental United States. So what does this mean? If I want to know how many more hydrogen ions there are, I would do this. You take the number 10 here from the base 10 representation and you raise it to the power of 3, which was calculated by doing this statement here, right? So I'm going to take this and bring it over as the exponent. Okay, and if you do that calculation, you're going to get this, right? 10 to the 3 represents 10 times 10 times 10. So 10 times 10 is 100, and 100 times 10 is 1,000. Okay, there are 1,000 times more hydrogen ions in Los Angeles rain than normal rain across the rest of the continental United States. That's what this means. That's what it means when they're, for every 1 unit change, for every 1 integer change on the pH scale, you get a 10-fold change in hydrogen ion concentration. So when we did our calculation here of 5.3 minus 2.3, that is 3 integer changes on the pH scale. So you take pH as it is defined as a base 10 logarithmic scale, take the 10 as your base, raise it to the power of 3, and that will tell you how many additional hydrogen ions are in Los Angeles rain. 1,000 times more hydrogen ions in LA rain than average rain. Okay, so I know this is not everybody's favorite topic, but I really do want you to think about this. I'm not going to beat you up with math, but I do expect that you understand, doggone it, that you do understand point number 7. Okay, that will be important to me and it will be important to you on an exam. For every 1 integer change on the pH scale, there is a 10-fold change in the hydrogen ion concentration. Folks, what questions do you have right now? Is there anything I can try to help clarify for you? Anything at all? Are you sure? This is not a topic that students traditionally do well with, and I'm just talking from experience. Okay, everybody's quiet. I don't know whether you're quiet because you understand or whether you're passed out on the floor. I don't know. So I hope you're okay. Let's continue at any rate. Okay. All right, folks. So we've talked about pH, and I think I'll let that rest for the time being. Okay, Israel, you're passed out on the floor. I hope you're okay. Ha ha, laugh, laugh. Good, good. I appreciate humor. Good. All right. All right, folks, let's continue. Let's continue. Now, the last major talking point that I want to spend some class time on here to finish up chapter 4 is to introduce and talk to you a little bit about the various types of organic macromolecules. Okay, that's really what the chapter finishes in. Okay, if we look on page 44, there's section 2.5. It's got a big bold heading that says Overview of Organic Compounds. And you know what? The next pages to the very end of the entire chapter, the next pages to the very end of the entire chapter talk about the organic macromolecules. Okay, the whole way out to the last page in my physically printed book is page 59. Okay, so we're going to talk about an introduction to the organic macromolecules. Okay, and there's only so much time I have, folks, right? There's some responsibility on your part that you need to spend study time on this. Okay, so let's talk about this topic. First off, what does the word organic mean? Well, if you talked about the word organic and you're shopping at the supermarket, at the grocery store, you probably think, well, organic means natural. And in that context, that would be appropriate, right? Natural, or perhaps, perhaps a vegetable that was grown without chemicals, or, any number of things like this. Perhaps a hamburger that was prepared from an animal that was grown without any antibiotics. So it's organic, right? Organic meat. Okay. That's appropriate for the shopping, for the grocery store. Organic here in science is going to mean something a little bit different and more precise. Organic means that the molecule contains carbon atoms. Okay. Now, I am not going to ask you to draw out chemical structures on an exam. I'm not going to do that. Even if we were together in the classroom taking an exam on a piece of paper, I would not ask you to draw that kind of thing for this course. Of course, if you're in a chemistry course, a biochemistry course, that would be a totally reasonable thing to ask students to do. But I won't ask it of you, but you still need to be looking at things and understanding things. For example, in figure 213, and I'll put this question in your notes, in figure 213, let me just do something here. In figure 213, how many carbon atoms do you count on the left molecule shaded purple? Go ahead and type your answer into the chat window. How many carbon atoms do you count on the left molecule that's shaded purple? How many carbon atoms do you count? Okay. Let's get more people contributing, okay? All right, awesome. There are 6 carbon atoms. 6 carbon atoms. Good job. Do you notice, folks, when you look at figure 213, that in the left molecule, you see the carbon atoms explicitly written out, but in the right version, you have a sort of chemical shorthand where you simply see the vertices of the polygon as the carbon atoms. Okay, so you're absolutely right. There are 6 carbon atoms. You all did it well. And my point is that whatever that substance is, right, it looks like probably a glucose molecule there. Okay, it's made of carbon atoms. As we page through the rest of the chapter, you're going to see similar things, right? All the molecules in pages 46 and 47, figure 214, figure 215, figure 216, are molecules that are assembled with carbon atoms. As I turn to page 48, figure 2.17, figure 2.17, look at any of the fatty acids there. Take a look at palmitic acid. Take a look at oleic acid. Aren't they basically chains of carbon atoms? Chains of carbon atoms. As I turn the page in the textbook, figure 218, showing you the structure of something known as a phospholipid. Just take a look at the atoms that are present. Don't you see a whole bunch of carbon? Figure 219, which is pointing out steroid molecules. These are rings of basically sugars that are glued together to form the steroids. Those rings have carbon atoms in. Page 52, figure 220 and figure 221 is showing you the building blocks of proteins, which are amino acids. Do you only see carbon atoms? No, you see other things, right? You see nitrogen, you see hydrogen, but you definitely see carbon. So carbon is present. If we continue this conversation to, let's go to page 56, figure 224, figure 225, you will notice structures that also contain a great deal of carbon atoms. So the long story here is that all the categories of organic macromolecules are described as being organic because they are all based off carbon atoms, okay? Here's the take home. Okay, all categories of organic macromolecules are manufactured by assembling, meaning by connecting many carbon atoms together. Many carbon atoms together. Okay. So why are the organic macromolecules important? Well, gang, there's a lot of things that can be said, but I'll say this. If you think about one cell of your body, most of its mass is made of water, right? 65 to 70% of the mass of every cell and of your entire body is simply due to the presence of water. Well, if we say water represents 70% of a cell, what is the 30% made of? The 30% is made of the organic macromolecules and things that are chemically related to them. So everything else that's not water is made of these categories of molecules that we call organic macromolecules. Okay, everything inside a cell that is not water is made of these organic macromolecules or related to these organic macromolecules. Okay, this is such an important topic that honestly, for students that are going to be full-fledged science students, or perhaps students that are going to go to medical school, there is a required course in undergraduate education, which is a 2-semester course called organic chemistry. It's a pretty intense course, but let's be honest, AMP here seems pretty intense too because of all the volume of information you have to get comfortable with. So, okay, these are the organic macromolecules. So let's first list out what kind of organic macromolecules there are, okay? There are 4 main types of organic macromolecules. Here they are. They are carbohydrates. They are lipids. They are proteins. And they are nucleic acids. Carbohydrates, lipids, proteins, nucleic acids. These are the 4 major categories, the 4 main types of organic macromolecules. And I'm just going to go through each category and tell you a little bit about each. But you're going to have to spend a significant amount of your own study time getting comfortable with these things. Okay. I will not ask you chemical reactions on the exam. That would not be appropriate because many of you haven't had a chemistry course yet. I'm not going to ask complicated chemistry. I'm not going to ask you to write out a chemical equation or a chemical structure, but it's totally reasonable for me to ask you to understand the vocabulary aspects of, well, what is a carbohydrate? What is a monosaccharide? What is an enzyme? What is a protein? These are appropriate questions for beginning students. So let's see what we can do here. Let's start talking about carbohydrates. Okay, so I really like, some of the tables in this textbook. Now take a look at table 2.6. It will tell you some of the main types of carbohydrates. Okay, so types of hard carbohydrates is going to include monosaccharides. All right, so an example of a monosaccharide would be glucose. That's arguably one of the best examples of a monosaccharide. Okay, but there are many monosaccharides listed in figure 2.14, right? Figure 2.14, and you should spend some time looking at that. Okay, so monosaccharides are the most fundamental, quote unquote, simple version of a carbohydrate that can exist. If you were to break down the glucose into even the smaller subunits of individual atoms, then we would not consider it a carbohydrate anymore. Okay, so a monosaccharide is your simplest type of carbohydrate that can exist. Which is why monosaccharides are often referred to as simple sugars. Simple sugars, right? All right. We also have, disaccharides from table 2.6. Disaccharides are shown to you in figure 2.15. So sucrose would be an example of this. Sucrose would be an example, figure 2.15. And I will point out that you might want to pay attention during your study time to these processes known as dehydration synthesis. Okay. Take note of dehydration synthesis and hydrolysis. Okay, I'm not going to define those for you right now. You define them for yourself. You do the studying. Okay, but be aware of these things. So long story short, if we take two monosaccharides and link them together with a covalent chemical bond, we build a disaccharide. Well, we could put three monosaccharides together, 1, 2, 3, and it would be a trisaccharide. We could put 10 monosaccharides together, and then we could name it an appropriate name for that, right? So we can begin to chain these things together, and eventually we can chain them into hundreds to even thousands of monosaccharides linked together. And when you do this, you'll get what's known as a polysaccharide. Okay, an example of a polysaccharide is going to be glycogen. Okay, glycogen is a storage polysaccharide found in animal tissue. Okay, the equivalent storage polysaccharide in plant tissue is going to be starch. And I say that because, I bet you some of you, particularly over Labor Day weekend, you might have had some French fries, right? From potatoes. Potatoes are plants, right? So these are various kinds of long, fairly complicated carbohydrates. You don't have to know the chemistry of these things other than you have to know what these words are referring to. Okay. All right. Let's see here. I like to get little pieces of pivotal foundational information into my students' memory banks, that things I really want you to appreciate. So I'm going to talk about something that I refer to as the basic building block. The basic building block is the most simple form of the category that can be used to build other things. And I've already told you what it is. The basic building block of carbohydrates is the monosaccharide. You take a monosaccharide, you link other monosaccharides to it, you build disaccharides, trisaccharides, so forth, till you get to polysaccharide. Okay, so the basic building block of this category of organic macromolecule is the monosaccharide. By linking monosaccharides together, we build other higher order structures. Okay, so what are carbohydrates good for? What does the body use carbohydrates for? Well, there's lots of things, and we could take an hour to describe that. I don't want to take an hour to describe that, but obviously energy, right? Things like sugars contain a lot of biochemical energy in them, so they're simply used to manufacture energy that can power the contraction of your muscle cells, so you can wave your hand, so you can walk across the street, so you can jump up in the air and catch a ball, right? So energy is a big use of carbohydrates. Carbohydrates are broken down to form ATP, right? We'll talk about ATP later, but you may know about ATP. Adenosine triphosphate is the biochemical energy, the biochemical dollar bill of the cell. So carbohydrates are definitely very influential when it comes to energy. Carbohydrates can also be used to build structures. Okay, plant cells, for example, plant cells will use carbohydrates to build something known as a cell wall, okay? So carbohydrates are not just for energy, but they're also to build things. They actually form structures, kind of like Lego bricks, where we're assembling higher order structures to build a carbohydrate wall called a cell wall around plant cells. Our human cells do not have cell walls. What plant cells do, bacteria that are in your body have cell walls. Fungi have cell walls. So many types of cells in the world, including some that are in your body and in terms of microorganisms, do have cell walls. Okay. All right. Folks, would you have any questions about carbohydrates? Okay, let's go to the next category. The next category is lipids. Okay, again, I'm going to stay with the organization of your textbook. Table 2.7 is very nice. It talks about types of lipids. And wow, this can get pretty involved here. So we'll say fatty acids. Okay, we'll say fatty acids. And so fatty acids are shown to you on figure 217 there. Okay, picture A shows you the example of saturated and unsaturated fatty acids, namely palmitic acid and oleic acid. So I'm just going to type in one example, palmitic acid. You see this in figure 217. Okay, you don't have to be able to write out the chemical structure, but you should be aware, if I were to show you a picture of any of these categories of molecules on an exam, I might ask you to tell me what category the molecule belongs to. That's fair game for the exam. Okay, so we have fatty acids, we have triglycerides. Okay, triglycerides are also shown to you on figure 217. Okay, so I'll let you look at that during your private study time. We have phospholipids. Which are actually going to be on the next page, figure 218 there. Okay. Okay, we'll be talking more about many of these things in the chapters to come in the course. Okay, steroids represent an important category of lipids. You know, maybe some of you have been on some kind of steroid medication to suppress inflammation in your body associated with a disease, or, you know, maybe you broke a bone and there's inflammation. So steroids are commonly used to suppress inflammation. Steroids are lipids. Our sex hormones, estrogen, testosterone, progesterone, are all types of steroids. Okay, these are all lipids, and we'll learn specific things about them as we get deeper into the course. We also have these beasties called eicosanoids, which are listed in table 2.7 there. Okay, eicosanoids are involved in many things. You can read it there for yourself. Blood clotting, stomach acid secretion, lipid breakdown. Okay. We also have what your book is calling other lipids. Other lipids gets a little bit vague, but as an example, you know, some of our vitamins are lipids. Includes some vitamins, some hormones. Okay. So let me give you the basic building block here. Okay. The basic building block for the lipid category, I would say, you know, lipids represents a very chemically diverse category. So it's kind of hard to give you one simple statement here, but the statement that I think fits most cases and is a good starting point for your knowledge is to say that lipids' basic building block is in fact fatty acids. Fatty acids are the basic building blocks of lipids. Okay. All right. So running out of time here. You know, what are lipids good for? Well, I can't give you an exhaustive response. There's just too much here. But I'll say a couple things. Okay. Lipids are used to form cell membranes. Okay, cell membranes. So that's going to include the phospholipid membrane of the outer border of the cell, but it's also going to include the internal membranes of the cell and what's known as the endomembrane system, which would include like the Golgi complex, the rough endoplasmic reticulum, the membrane of the nucleus. These are things you are going to encounter in chapter 3. We're almost there. We're almost there. Israel, the answer to your question is, Israel's asking, do we have a practice test today? Israel, there will be a practice test given in lab later today. Yes, the practice test is just for practice. It does not count towards your grade. Okay. So lipids are very useful for membranes. Let's see here. Some lipids are used for communication. Okay, and by communication, I'm referring to lipids that are acting as hormones. Some lipids act as hormones. Okay, so lipids have a variety of functions in the body. Okay, folks, it's 9:15. I've run out of time on this, so here's what I'm going to do. Okay, I'm going to momentarily stop. And what I will do is we'll take a break. We'll start our lab. When we start lab, I'm going to finish this conversation in your notes. I'm going to give you additional notes for maybe 15 minutes so that I can finish chapter 2, so that next week, Monday, I can and will start chapter 3. So we'll start lab by finishing our lecturing here in chapter 2. Then we'll go into the work we have to do for lab this morning. Folks, let's take a nice break. Let's have lab start at 9:35. So let's take a 20-minute break. Go. The Zoom channel will stay on. You don't have to log off. and I'll see you at 9:35. Okay. All right. So go ahead and take a break.
Thank you.
Israel, all the notes are on Wiley Plus. Yes. Okay. See you folks in about 20 minutes. Okay, welcome back, everyone. And let's see if we can reconvene here and get our momentum back. So before I finish our notes here in chapter 2, is there anything you'd like to ask? Okay, all right. So let's do that, folks. Let's finish our conversation describing the different categories of organic macromolecules. I'm going to share my screen with you. All right, so we've defined carbohydrates and lipids, okay, and I've referred you to some of the pictures in your textbook. Let's go to the next category. So this will be number 6 in our list, which are the proteins. Okay. Now, for carbohydrates, there's different types of carbohydrates like monosaccharides and polysaccharides. For lipids, there's different types of lipids like fatty acids, triglycerides. For proteins, there's not really different types. Proteins are simply proteins, okay? So no different types of proteins exist, but proteins do perform a variety of functions in the body. And we'll list that out in just a bit, okay? So proteins are talked about, at least beginning with page 51. Okay, well, okay, I see what your book is doing here. Let me walk that first statement back and say, okay, there are different types of proteins. Let's take a look at table 0.8, table 2.8. On page 51. I wouldn't typically organize the information this way, but we can do this. We can stay with the organization of your textbook. Okay, there are proteins that are structural in nature, okay? Your book gives you collagen as an example there. Okay, collagen is one of the major proteins for a lot of structures like bones and hair and things like this. We have regulatory proteins. Okay, we'll give you some examples of these in just a moment. We have contractile proteins. If we're thinking about contraction, think about your heart muscle, skeletal muscle, etc., etc. We have proteins that are involved in immunology, so immunological. Okay. Your book specifically talks about antibodies. Okay, we can do that. So, if any of you did get the COVID vaccination, right, one of the things your body did was manufacture antibodies. Antibodies are proteins that function in the immune system. So we can talk about them in this kind of context. We have transport proteins. Okay, so there's many proteins involved in transport. So your book there in table 2.8, page 51, talks about hemoglobin. Hemoglobin is not strictly a protein, it's actually a compound, but I understand why your book is saying that. Okay, it is used to transport blood gases. And then we have, according to how the information is organized in your book, catalytic. And when they say catalytic, they're talking about enzymes, okay, enzymes. Enzymes is a very important category because enzymes accelerate chemical reactions. They make the chemistry of your body, the chemistry of your cells, the chemistry of your metabolism happen quicker than it would without the presence of the enzyme. Okay, so these are all very useful things, and typically we talk about them in more of a focused way when we're talking about a specific topic. Like when you talk about the respiratory system in AMP2, you'll talk about the specific functions of hemoglobin, okay? All right, so this gives you an example of the various types of proteins as organized according to the textbook authors. As I said, I would probably do it different, but I'm going to stay with your textbook so you have that resource and are not confused. The basic building block of proteins, without a doubt, is your amino acids, okay? There are 20 naturally occurring amino acids. Your book does not list them all out. But it does provide some examples of amino acids in figure 220. Okay, just like with carbohydrates, you could take one monosaccharide and link it together with another monosaccharide to build larger structures. Amino acids are like that. They can also be linked together like Lego bricks. One amino acid linked to another amino acid to another amino acid, and you can build large proteins with it that do a variety of things. So yeah, let's get to letter C here. What are proteins good for? Well, many things. Okay. Okay. We'll start with enzymes. Okay. Some proteins make chemistry happen quicker. These are the enzymes. Okay. And so if you drink a glass of milk or you have milk with your cereal, right? Your body's got to digest that milk sugar, which is called lactose. So your body has enzymes called lactase, which will actually break down the milk sugar. Okay. So lactase enzymes break down milk sugar known as lactose. Okay. Let's see what else we could say here as an example of proteins. Okay. Let's talk about more transport functionality, right? The book mentions hemoglobin, but let me give an example. Proteins are used to bring cargo into cells. I'm specifically thinking about glucose transport proteins. Some cells are used for binding of cargo on the some proteins are used by cells to bind cargo on their outer surface. So like if you had a molecule that was put into your blood from food that you've eaten and digested, that molecule might bind to the surface of the cell and get into the cell by attaching to a protein. Okay, so what are proteins good for? Well, binding cargo outside the cell and bringing it into the cell. Okay. Other proteins are used to regulate chemical processes. I'm thinking specifically here about insulin, right? Insulin is going to be the hormone that regulates sugar breakdown in the body. Okay, so there's 4 quick things illustrating some things that proteins actually do for us. The list is basically endless, and it's exhaustive. I mean, we could not literally enumerate everything. It would be too much for our brains to hold. Okay. Let me just make a comment about protein structure. So I'm going to make this bullet point letter D here, right? So remember in chapter 2, last conversation and today's conversation, we made a pretty big point about saying that changes in temperature will change the shape of molecules, which change their function. Well, when you're talking about changing the shape of a molecule, you're talking about changing the structure of a molecule. Now, typically, we don't talk too much about the structure of carbohydrates, lipids, or even nucleic acid in this course or other courses. There's a lot of reasons for that. But suffice it to say, I think we can honestly say of the 4 categories of organic macromolecules, which category do we scientifically understand the best from a structure perspective? That would be protein. Okay, we probably have the deepest scientific understanding. Mankind probably, arguably, has the best understanding of protein structure than it does carbohydrate, lipid, or nucleic acid structure. So let me just mention the 4 levels of protein structure. And this takes you to figure 222. Okay. I will need you to spend some time looking at this stuff. But the point is that these different levels of structure work together to form the overall shape of a protein, and the overall shape of a protein defines what that protein is able to do for the cell. Okay. All 4 levels of structure interact in ways to produce the final shape of a protein and give that protein its function. Okay, so the 4 levels are, primary structure. Okay, you can see that that refers to literally the sequence of amino acids. One amino acid, next amino acid, next amino acid. Scientists have for a very long time been trying to write computer programs that could take the primary sequence of a protein and use that to develop the shape, the geometry of the protein in the computer, to computer model the protein. That's been a very hard task that's been worked on for over 50 years. And there's been a lot of setbacks and frustration. But recently, a company owned and funded by Google had some fantastic results using artificial intelligence techniques and machine learning techniques that they've gotten some phenomenal results predicting the structure and shape of proteins based on the primary sequence of a protein. This is only interesting to those of us that admit being geeks, but it has practical relevance because a lot of disease processes take place when proteins malfunction. The malfunction is associated with an abnormal shape. So if we know what the shape of these things is supposed to be, it gives us a way to start thinking about how to clinically treat it to help somebody. Okay. All right, we have secondary structure. Okay, the secondary structure refers to these things known as alpha helices and beta pleated sheets. The secondary structure comes about through interactions of amino acids in the primary sequence. That's what's taking place here. Okay. We also have a tertiary level of structure. Okay. This refers to an interaction between the alpha helices and beta pleated sheets. Tertiary structure comes about from interactions at the secondary level. Okay. And the quaternary structure is not present in all proteins. Okay. Okay. You will only find quaternary structure in proteins that are actually made of multiple fragments of amino acids called polypeptides. So it's kind of like having multiple small proteins locked together that gives you the quaternary structure. All right, so don't worry too much about that, but at least since your book gave it a full page description, something that I could draw vocab from during a typical exam. All right. Gang, do you have any questions about proteins? I know this is a lot of content, right? It is a lot of content. Okay. All right. Thanks for the feedback, Shay. All right. So let's finish our conversation by talking about nucleic acid. That's number 7. Okay. We have types of nucleic acid. And so for this, there really isn't a table in your book, but I can give you some types. I would say that there's basically 2 types. There's DNA, which is short for deoxyribonucleic acid, and RNA, which is short for ribonucleic acid. Okay. The basic building blocks. So if you want to look at some pictures of DNA and so forth, you'd be looking at figure 224, 225. Even figure 226 has relevant information here. The basic building block of nucleic acids is something that is called a nucleotide. And what a nucleotide is made of is defined for you in figure 224. Okay. You don't have to be capable of writing out the chemical structure. I keep saying that. But you do need to spend enough significant study time so that the word nucleotide is not foreign to you and you know what it's referring to. Okay. All right, so let's see here. What are nucleic acids good for? Well, okay. If we're talking about DNA, DNA is an information-containing molecule. That basically provides all the instructions for a cell to be the cell it is and to function and be alive. In other words, the DNA contains the blueprint of life inside a cell. It's basically an instruction manual for your cells to be your cells. Information-containing molecule that provides information to cells about how to function and what to do. Okay, so that's, DNA and RNA are the molecules that allow life to exist on the planet as we understand it. Okay, RNA. Everything I'm saying here is just a glimpse of the complexity awaiting a student for a lifetime of study, right? I might know a lot, but I'm not done and I will never be done, right? Nobody ever finishes knowing this stuff because there's so much to learn. RNA is a middleman, if you will, between the instructions of DNA and the production of protein. Okay, RNA is used as sort of an intermediary molecule to help cells know when and how to manufacture proteins that then go off and do other things in the cell. Okay. All right. So lots more can be said about each one of these topics, but as you can see, things are already getting long in our time together this morning. All of these categories, we will encounter specific cases of them being involved in specific processes in your body as we continue through the semester. We're just getting started, folks. This is not done, okay? So is there anything about the organic macromolecules, water, pH, covalent ionic bonds, anything from chapter 2 you would like to ask about? Because I am going to start chapter 3 on Monday. Okay, well, please let me know if you do have questions. Remember, do the practice questions in your textbook, do the figure questions, critical thinking questions. I know you have a lot going on. I'm assigning a lot of homework from lab, but you must find time every day to study for this course. So Shay, you ask, are inorganic important for us to know? I would say yes, Shay, but when you say inorganic, you're simply referring to things that aren't made of carbon. So let's take an example of something we talked about in chapter 2 that is inorganic and which was important for our conversation. An example would be sodium chloride, table salt. We talked about sodium chloride, table salt, to help us understand ionic bonds. Sodium chloride is an inorganic substance. So yes, I would say that's important for you to know. Okay. Okay. Is there anything else you'd like to ask about? Brittany, I do see your question to me, and I am aware of your email. I'll get back to you later today, I promise. Okay. Anything else? Okay. All right, gang. So what I'm going to do is stop sharing my screen momentarily. I'm going to stop the recording because that completes the lecture.